Okay, guys, we are going to be getting into genetic mutations. Now, we all know that mistakes happen. This is very normal. And it can sometimes even happen in your cells. So these mistakes can have a range of effects on the body. So when they occur in DNA or RNA, this is when they are referred to as mutations. Now, a mutation is a change in an organism's nucleic acids, so in RNA or DNA. But there are two types of mutations. You have gene mutations, which occur in genetics, and you have chromosomal mutations, which is in regards to heredity. Now, first, we have a point mutation, which is also known as a substitution. So, a mutation in which one nucleotide is put in place of the correct nucleotide. Usually, a mistake like this is caught and fixed by DNA polymerase, which, if you recall, is the proofreading system that DNA has in place during DNA replication. If not, the substitution may permanently change an organism's DNA. So an example of this is known as sickle cell anemia. So here we have our original sequence of DNA, and we know this is our DNA because of the presence of the T. Now we can see that a point mutation has occurred. So instead of there being a T here, it has been substituted or replaced with this C. Now another kind of mutation we have is a frame shift mutation. So this involves the insertion or deletion of a nucleotide in a DNA sequence. So this shifts the entire sequence by one or more nucleotides, which tends to have a bigger impact. And an example of a frame shift mutation occurs in cystic fibrosis. So let's say this is our original DNA sequence. You notice that we have had an insertion right here of a C, which has then moved the rest of these down, meaning that the production of amino acids from the codons is going to be changed. So here's another example. We have our original sequence. And now we have a deletion, so it's moving everything to the left to replace what has been lost. So as you can imagine, the beginning of the sequence here is going to be the same, but once we encounter the deletion or the insertion, that is going to change the amino acids that are going to be produced based on the codon frame shift, which means that mutations that are or are not supposed to be produced is going to change the entire protein production of that sequence. So here's another way to look at it. I'm going to give you this short sentence. The dog sat. But imagine you took out the H in the in this sentence. So, Ted Oggs at. That doesn't make any sense because we've taken out the H, and since we know that codons are read in groups of 3, we had to move the D over. So this is an example of the deletion when it changes the reading frame, which results in the codons that might code for different amino acids. So once again, the dog sat. But what if we inserted something in between our H and our E? We put a T in there. And you may try to say that. I really don't want to. But this is an example of when the insertion changes the reading frame, which once again will result in the codons that might code for different amino acids. So mutations may or may not affect an organism. An example of this is that suppose a substitution occurs in a coding region of DNA that changes an AGG codon to a CAG codon. So AGG codes for lysine, which helps enzymes bind to its substrate, while CAG is glutamine. So the substitution could affect the protein folding and destroy the protein's function. So this is known as a Mutations mutation. So suppose now we have a substitution occurring in a coding region of DNA that changes a UCA codon to a UAA codon. So UAC codes for tyrosine, while UAA is a stop codon. So this substitution could result in a premature stop codon, and this is known as a nonsense mutation. Now, remember that some codons code for the same amino acid. So let's suppose that an A is substituted for the G. So let's look at AAG, which once again codes for lysine that helps bind enzymes to its substrate. But now we have AAA. This also codes for lysine. So this substitution could have no effect. So this is what we call a silent mutation. Now, mutations are caused by several different factors. You can have replication errors, errors in mitosis or meiosis. We can have errors in protein synthesis, or we can experience mutagens. Now, mutations are not uncommon. So examples of mutations, we have some that are considered harmful, like cystic fibrosis, some that can be helpful, like people being born with an immunity to typhoid fever or other kinds of illnesses. And then we have some that have no effect, like red hair. Red hair is not an uncommon genetic mutation, but does not have a specific negative outcome on the person who displays this mutation. Organisms have many tools to help them. So DNA has a built-in proofreader, which is known as DNA polymerase. Sometimes the DNA polymerase can miss an error, and this is when a mutation occurs. Now, examples of mutagens can be ultraviolet or UV rays from the sun, industrial chemicals, carcinogens, or infectious agents such as viruses and bacteria. Now, by introducing these mutagens are basically known as outside factors that can affect the DNA of an individual. Now, that's it for mutations and mutagens. Make sure on your information sheet you are filling in extra information that you feel like is important for you to understand mutations. And we are going to be hitting this a little bit heavier in our next